Hi everyone. This time we're going to move on to session 4 and 2, which is about the next generation sequencing. This class will cover basic introduction of what is the next generation sequencing and particular focus on Illumina sequencing, which is one of the dominant technology we can use in biological research. And then we compare the differences between single-end and paired-end sequencing. Then after that, we are a little bit discuss on what is the long-read sequencing technology. And after that, I'm going to introduce the base calling method, then compare the types of different genome sequencing. So let's start off the what is the genome sequencing, right? So as I mentioned last time, sequencing is the process of determining the nucleic acid sequence, right? And so we basically try to find out the order of DNA nucleotides in our genome. So sequencing is basically get to know about what is the nucleotide composition in your DNA sequence. But in a different context, we can also use knowing the nucleotide sequence in RNA genome, like a virus or RNA expression, that also we use the genome sequencing as well. So genome sequencing is basically we get to know about the nucleotide sequence, whatever in DNA or RNA nucleotide in our genome. And basically, this is the activities on the reading sequence, right? So what we get to know is genotypes or allele at a certain locus and changes in nucleotide sequence compared to reference genome. So we call the variant, right? So as we discussed in the previous sections. So genome sequencing is basically the basis of finding a link between the genotype and phenotype. So that's why it's quite important technology we can have in our biological research. We specifically call genome sequencing in these days is next generation sequencing. Shortly, we say NGS, right? Why we call this is the next generation sequencing, as we learned last time, we have the previous generation of the sequencing, which is the Sanger sequencing. That is the first technology we can use for to find out the nucleotides in our genome, but that is quite laborious and very low output of the data set. So people trying to advance this technology, get more data set. So in these days, we say it's in high throughput or ultra high throughput. So, and also this is sort of consecutive advances, to Sanger sequencing. So we say it's a second generation sequencing. And also, since we get a high throughput data set, we also call this is massively parallel sequencing. So first NGS introduced around mid 2000 by Roche, the technology platform called 454. And at that time, there's another alternative we call Solexa technology. Later is M&A by Illumina sequencing. So this is sort of prototype of Illumina sequencing at the time. And afterwards, few more technology coming into the field. So what is basic principles of NGS technology? NGS sequencing is basically produce a sequence read. So when you know about the DNA sequence, we say it's a sequence read, right? So sequence read is a fragment of nucleotide reads by sequencing technology. And NGS mostly produce short-read sequencing, especially the Illumina sequencing. So once you get the certain fragment of your DNA, is basically you break down your 3 billion base pair DNA sequence into the tiny, tiny pieces. Then you use those fragments, then you get a short-read information. And once you get these fragments, then you add on the adapters on these fragments. So we call this is the library, right? So library is the DNA fragments apart from the genome sequence. Then you get the adapters at the end of that. Then you put it into the sequencing, right? So once you have those fragments or library, literally this means that the pieces of information, amongst the entire DNA sequence. So we have the partial information. So we have the short reads, we call the short reads of your DNA sequence. And once you get those information, we do the sequencing by synthesis. So we generate the nucleotide information out of your library. We'll get to that point in the later. Ancient read sequence is around 50 to 300 base pair, and it really depends on the technology. So you sometimes have 50 or sometimes have 300 base pair. And of course, the technology, they will, improve their limitation, which is the short reads. So they evolved to produce longer reads. So ultimately, it's settled down into the long read sequencing introduction. And more sequencing outputs than single sequencing. That's obvious. That's why we call this is high throughput sequencing or massively parallel sequencing. So what is the throughput? It's the amount of output. So how many DNA information you get from those sequencing. So let's say single sequencing, you get only 100 base pair out of the one single sequencing experiment. But if you do the next generation sequencing, then you get a 90 gigabytes information. So that's much higher than that. And depth, as we discussed last time, if you want to read certain area, how many times you read that, right? So you read that information. So you read that information. So just reading one time is not a very good thing. So you get 10 times, you get 30 times, you get 100 times. So you get a more confidence out of this information, right? So this is an important thing. And you get a coverage, right? So you get a 3 billion base pair of your entire DNA sequence, and some of the area is not easy to be sequenced, right? Because regions around centromeres or telomeres, those are very complicated sequence information. So those regions not really covered well. So coverage is literally how much regions you really covered by your sequencing activity, sequencing experiment, right? So ideal situation is 100% coverage, you cover everything, but real time is not, right? So this is one of the aspects you have to consider in next generation sequencing. Okay, so let's talk about the Illumina sequencing. So this is basically the sequencing by synthesis. So you firstly sequence the nucleotide information by chemical synthesis, right? And where the synthesis happen, it's on the flow cell. We'll get to that point. So flow cell is like a solid surface, it's like a tiny chip on like 2 fingers. And onto this solid surface, you put the library into those areas. So this is one very tiny spots of the flow cell. Onto this flow cell information, you put the library onto the priming station. To this individual spots we call priming station. Then you put the library like in here, then you put in here. So we'll get to that point in later, but a little bit shortly explain about that. So this is the library scheme. So you have the 2 adapters at the end of the DNA fragment, and this is the DNA fragment, right? So once you chop up your 3 billion base pair DNA sequence into very short reads, short fragments, then you put the adapters at the end of that. Then these adapters used to bind to the priming station. So this is the little scheme on that. Then once you get the priming station binding, then you get all that information happen within each stations like in here. So many wells, many spots, right? So you get a large amount of information out of this area. So once you get that information, you through the synthesis for your sequencing. For that purpose, you have to use the fluorescent dye. Then that information can lead you to which nucleotides bind to certain data points. So Illumina sequencing, once you get the library bound to the priming station, then you use another primer at complement to other adapters here. Then you attach to each nucleotide onto here. And each nucleotide is synthesized with the fluorescent. Then once you get the synthesized, then fluorescence gets emission of the light. Then this emission light will be captured by imaging by the camera on the machine. Then you get to know about the nucleotide information. Then once complete those information, then you cleavage this fluorescence, then you go through the another rounds of synthesis afterwards. We'll get to the point after that. Okay, so Illumina sequencing, there are many different competing NGS technology, but Illumina is for this stage around 2024. This is the best technology in terms of the price, in terms of the throughput, in terms of the usability, in terms of the coverage. So people use Illumina most. Illumina has the 4 basic steps. So first one is DNA and library preparation. Once you extract the DNA, you put it into library format. Then once you do that, then you throw out the fluorescent flow cell after that. Then you do the sequencing. Then after that, you do the bioinformatics analysis, right? So let's start from the library preparation. So library preparation has this basic process. So first thing is you first fragment the DNA. Then you repair the ends, so ready for the adapter ligation. Then after adapter ligation, then you do the DNA selection. So first step is the fragmentation. So how we make a DNA fragment out of the 3 billion DNA sequences. So since the DNA sequence is really long linear sequence, then you have to chop up that DNA sequence into the pieces. So how can we do that? We use the physical force, which is the ultrasound waves. So you put the DNA into the water, like a DNA, that is a soluble material. So you put it into the water. Then within this water, then you put the ultrasound wave. Then you physically breaking down those DNA structure into the small pieces, right? So since this is the physical force, up to the, how long you do that process or how many times you do the process or how, far you do the process, your output will be different, right? So if you look at here, you do the 13 cycles, then your DNA sequence, your DNA will be 200 base pair fragmentation, right? Around the 200 base pair. But if you do the only 5 cycles, that will have like a 350 base pairs because it's less final, because you put less force into the fragmentation. So it really depends on how much force you put it into your fragmentation, then you get the different result, right? So why we are doing this fragmentation? Because we want to put the ligation, adapter ligation at the end of the each fragment, right? So basically, once you synthesize the DNA sequence, like in nature, you use the phosphorylation ligation. So like in here. So if you have the fragmentation, whatever you do in the replication situation, you always use the phosphorylation at the end of that, then you add up the new nucleotides on that. So this is, exactly same we used in this library preparation. So once you get the fragmentation, then you make a blunt end repair with the phosphorylation, then you put the annihilation on here, then you're ready to put the adapters at the end of this ligation. So if you look at on here, you will get this adapter sequence like in here. And if you use this adapter sequence, then you can do many different tasks after your sequencing procedure. So what we can do, adapters will bind to complementary oligo on the flow cell, as we see in the previous sections. And also, you can do piece enrichment of adapter ligated DNA fragments. Then you can make sure that you use the DNA fragments only with the adapter sequence. Then you can fully utilize this library into the sequencing purpose, right? And also after that, you can use the index or barcodes of the samples. Then you can allow to multiplexing, or you can allow to many samples into the same experiment. So that's basically why we use adapter ligation in our experiment. So firstly, we see the index or barcode sequence first, right? So we have the 2 adapters we used in the adapter ligation. Especially the P7 adapters, it has the 6 variable nucleotides, which I represent N. N can be everything, like an ATGC, right? So you use these barcodes to make a multiple samples into the same experiment. Why we need that? So for instance, if I sequence myself, if I sequence three more students from the class, so we sequence four people, including myself, right? So if you make a library, then if you put it into the machine, if you put it into the library of these four people, the machine cannot tell, which samples are from, which person, right? So we want to make sure that we give the barcodes to each samples, right? So myself, I have AT, and the student one has the TT, and student two has CT, and student four has GG, right? So we all have the different, barcodes of certain nucleotides, right? So if you use this barcode information, then you do the sequencing, you get a four different sample data, then you can easily find out these samples belong to this person, right? So this is why we use the barcode. This is very simple example, but let's say, if you do the sequencing like 1,000 microbiome, samples, then you can use these six variable nucleotides as barcodes, and you can distinguish these 1,000 different samples, right? Why? Because how many combinations you can use for this barcode is 4 times 6, right? So it's too many things, right? 4 to 6, right? So this multiplex approach using this barcode is super useful. Then you can, affordably do the sequencing for high throughput situation. So that's why we use the index sequence. Okay, so P5 and P7, this is the complementary sequence then that combines to the at the end of the DNA fragment, right? So this is the DNA fragment prepared by phosphorylation and adenylation. Then it binds to the adapter sequence P5 and P7 in here, and P7 and P5 at the end of the other ends, right? So these two ends, both ends will have the adapter sequence, then you use that information, right? So one ends, P5 start from here, and P7 end, and P5 to P7 end, right? So 5 prime, 3 prime, 5 prime, 3 prime, right? So this is basically the structure. Then once you get the sequencing, you're not going to do the double strand. You're going to use the single strand, right? So the nature these double strand into the single strand, then you put it into the flow cell, right? So this is basically we use for the library sequencing. Okay, so then once you make this library, you do the PCR enrichment. Why we use the PCR enrichment? Because as I mentioned that you have the DNA library on here, and this is the adapter sequence on here. So this is the fragment. This is the fragment, and this is the adapters, right? But some of the fragments, they fail to have adapter ligation. So they have no adapter ligation, right? So if you have this amount of your sample, you cannot see that, right? Because this is in the water, right? In a reagent. So you put it into the sequencing, then half of the sequencing done by this one and half of done is not by this one. So it's waste of your data. It's waste of your experiment, right? So you want to make sure that you're going to enrich those adapter ligated fragments into many proportions. So after your PCR, you're going to have, let's say this is the 50-50%, but after that, after your 10th cycle, you exponentially increase the proportion of these, ligated library, then you get a most of the proportion with these libraries, right? Then your sequencing will be super useful, right? So this is basically why we use the PCR enrichment for library preparation. But some students might have some questions like, professor, you can do the, ligation efficiency up to 100%. 100% is not real. So let's say ligation efficiency currently is an 80%. So we see 8 to 2 proportion of this situation, but you can advance your, kits to make a efficiencies like a 99.9%. Then you won't think about this, unligated situation, right? And this is true, right? So these days, kits are quite, advanced, improved over the time. So efficiency is nearly 100%, right? So you don't really require PCR enrichment. But, just for your reference and also a little bit different library preparation, not like a genome sequencing or RNA sequencing. Some of the libraries, like a epigenome, we still require the PCR enrichment. So I briefly mentioned about this process into this lecture. Okay, anyway, so amplification, we use this primer information within these adapters. Then we use that information, then we can easily make a PCR amplification. Okay, so that is the amplification process. So once you get all the library preparation, then you can check whether your library preparation done correctly or not, right? So you can use the machine like an Agilent bioanalyzer. You use like this chip. This is different from flow cell. So you put the samples. How many holes on here? It's a 12. Then you put the samples on here. So you can basically put the 12 samples on here. Sorry. It's 12 samples, but here is some markers here. So it's a 16 wells, sorry. So but anyhow, you can use 12 samples on here. Then each sample, it will give you this graph, right? So graph is basically cover the library size from the 35 base pairs to 10K base pair. Then this is the density, right? So how many samples has certain proportion at certain range. So what is the peak of these samples is around 323 base pair, right? 323 base pairs. So most of the samples you put it into here. So samples here, library is the 303 base pairs, right? So what is the DNA fragment size? It's not 323 base pair. This is important, right? So adapters is around 75 base pair, if I remember correctly, right? So as I mentioned, this is the DNA fragment. Then here is the adapters at the end of that, right? So each of them is a 75 base pair. So adapters at the both ends is 150 base pair. So your DNA fragment size is 173. Okay. So this is the size of DNA fragment plus library. Okay. So this is the one. We have the sharp peaks on 35 and 10,380. Why? This is the marker, right? So Agilent bioanalyzer not running randomly. It has to run within a measurable range. How we can define that? They use their own ladder and they put the 35 and this range. Then they can check the known position, right? So within this position, you have samples around here, right? If the PCR is not working correctly, and if the adapter ligation is not working correctly, then you're going to have like this information. So 75, you have the sharp peak, and you have red numbers on here, right? Because these samples are not ligated, so this is shorter than that. Then 75 base pair, sharp peaks on here, because adapters, they forming much of that because they fail to ligate to the fragment. So they exist alone. Okay. So this is, we can check whether the amplification or whether the library preparation done correctly or not. So this is the thing. Another thing you have to keep in mind that, as I mentioned, that the PCR enrichment, PCR amplification, we're not doing these things in these days anymore because the adapter ligation efficiency getting better, right? And we don't need it. And especially if you have the PCR, then you have the same library many times in your samples. We call this is the duplicate reads. And this information we don't need anymore because this is the sort of the very redundant information. So you not care about this information much in your bioinformatics analysis, the parts later. So we generally remove those duplicated, the redundant information in the bioinformatics analysis. So if you do the many PCR process, you have the high chance of having this redundant information. So these days, when you prepare the library, you are, avoiding those PCI process, and you choose to increase the adapter ligation efficiency, right? Okay, so that is the thing you have to remember. Once you get the library preparation done, then you do the flow cell preparation. So flow cell, as I mentioned, that this is like a 2-finger size, solid surface thing, and this looks like this tiny plastic. And this has the 8 lanes, 1, 2, 3 to 8. And this is like a literally swimming pool lanes. And you put the samples, this tiny hole onto here. Then you flow like a swim to the end of the lane. Then you flow out to here, right? Flow in, flow out, right? And you flow in the library. Then some of the library, they're sitting around the stations, because it's randomly, they binds to complementary sequence on here. Then you get to all the library bounds to run here, right? So this is basically how flow cell is working. And also one more thing you have to keep in mind, not for exam, but just for your career. Flow cell has the barcode, right? Because barcodes, they recording where these kits from and where these experiments done and who's done that. And also this data used for which batch, right? So all of that information recording by this barcode automatically. And this is efficiently managed by laboratory management system. We call the LIMS or LMS, whatever. So this is all the automation. So this is enabling automated process out of that information. And this flow cell also has an ID, which is like this character, 1, 2, 3, 4, 5, and character on here. And this information, there will be stored in the data as well. So when you do the bioinformatics analysis, you get to know about which flow cells they data from, right? So this is also the things you have to keep in mind. Okay, so once you get the flow cell, it looks like this. So this flow cell priming station, if you look at horizontally, it looks like this. So this is a flow cell and the binding stations on here. And each library, they binding to complementary to here throughout the adapters, right? So once you get the double strand one, you have to separate that. But before you separate out, you first do the bridge amplification, right? So literally amplification throughout the bridging, right? So why we need it, right? So you basically put the one library on here, one library on here, one library on here, one library on here. You not put the another library just next to other library. You sparsely put on here. Then once you put the sparsely, then you put the bridging application, then you will get like these clusters, right? So this is the library 1. This is the library 1's cluster, and this is library 2. This is the library 2's cluster. This is library 3. This is library 3's clusters or something like that, okay? So why we need it? You basically amplify, you basically enhance the signals out of these clusters, right? So this is the requirement. So what is the examples? So once you get that information, so this is one of the representation. So this is the 1, 2, 3, as I mentioned. And for the simplicity, I remove out all the clusters, just put the representative 1, 1, 2, 3, right? So once you do the actual sequencing, we are throughout the synthesis, right? So what we're going to synthesis. So we use the nucleotides with the terminator and fluorescent and ATCG. And as you know that, it will synthesize through the complementary way, right? So you have the G, you synthesize on this position is G. One thing you have to note that the actual sequencing actually this way, not this way, but just for the simplicity and the legal examples, I put like this one. Okay, so this synthesis and C sequence, then you have the G, and you have the G and C. And once you've done that, you have the fluorescent emission, then you measure by imaging. So if you look at on the top, you see that this position has C, this position has C, this position has G, okay? And you remove this terminator with the fluorescent, then you do the another synthesis, right? So second cycle. So first cycle done, terminator, boom. Then you do another, add on to the sequence, okay? Then you capture that image as well. Then you capture that image as well. So why we need bridge amplification? You basically boosting the signals to detect those regions, right? So you have the many same molecules around here. Then if you go throughout that information, then you have the clusters of the same molecules on here. So once you detect the camera imaging take, then that will give you better signals than before, than just a single thing, right? Okay, so once you get the cycle, then you can easily infer that which sequence comes from here, right? So this one is the C, the complement is G, so you can read out the GATACCC, right? So this throughout that. Okay, so that's basically how we use the library preparation, then use the cluster generation, then you actually do the sequencing, okay? So this is the image of the fluorescent taken by camera, right? So you use that within a 20 micron, then you see that information like in here, right? So once you get those information, then you can detect this position has which nucleotide, right? Some of the things you cannot really see like in here, right? So this is the cluster, bridge amplification was not done correctly, right? So this is why we need that bridge information. Okay, so once you get the information, then you represent by the quality, right? And when you represent by quality, we use the Fred scale score. This is super important things because anything you represent the quality in an NGS, whatever base calling, whatever the variant calling, we always use the Fred scale. So Fred scale, as I mentioned, that this is the quality information, right? So how we can say good quality? Let's say you do the sequencing at a certain position, and the error you can have that one is the 10 to 1. If you proceed the 10 times, you have at least one time. Then that is your quality. Then some point you can have that you try 100 times, you get one error. Or you can do 1,000, you can have one error. Which one is good? This one is good because it has the less error, right? So you can represent your quality measure like this, denominator. But this is not a great idea to representation in terms of the simplicity, right? So you can basically use the Fred scale scores. How we can do that? First one is you basically do minus log 10, okay? So if you do the minus log 10, if you put this one, what is that? It's 1, right? What is this? It's a 2. What is this? It's a 3, okay? Then 1 to 3 is tiny interval, so you basically times 10, okay? So this is the Fred scale, okay? This is the Fred scale. So this is going to be 10, 20, and 30. So if you look at here, it's from 0 to close to 40, okay? So you have 10, 20, 30. What is that? 10 to 1 times you have an error, 101 error, 1,000 error, right? So can you see that the first part of the sequencing, which is the base pair position, at the very first few positions, they have good quality, and later it has the lower quality. We can easily interpret that, okay? So this plot is super important things to, understand the, sequencing by synthesis. So as I mentioned previously, we do the sequencing by the synthesis, and each synthesis we call cycle. First cycle, second cycle, third cycle, up to 6 cycle. So how many base pairs you reach on here? 6 base pairs, right? But if you look at here, oops, sorry, if you look at here, you get 300 base pair out of your library, right? Which means that you threw out the 300 cycles. Okay, that's an important thing. So how long one cycle takes? Let's assume that 10 minutes. If you take the 300 cycle, it will be 3,000 minutes, right? It's around 50 hours, right? And as I mentioned, that all these process is by enzyme, is by chemistry. So your chemical compounds going to drop out the quality because chemical enzymic reaction is not always 100% accuracy. It's viable at the very first time, and at the end of that, it has the less quality. So that's why the quality is dropped on here, right? And another thing is it's just a tiny information, not really important. Some of students might ask that, Professor, why these, first few cycles, they have less, quality than these steps? Because this process, they adjusting the cluster position, right? So they don't have the prior information. So they adjusting the quality at this process. So that's why it's artificially a little bit lower quality than the later parts. Okay, so that's the basic thing. And also one more thing you have to remember is once you have the library, like in here, as I mentioned there, this is an adapters, right? Adapter, okay? And the entire things from adapter to adapter is the library, okay? And what is this part? This part is the DNA fragment. A fragment, okay? Right. And if you do the 300 base pair cycles to know about the base pairs, we call this as an reads, right? Where the reads locate, reads on here, reads on here. Think about the priming station, right? You bounce to the priming station on here, then you read out here. So read out 300 base pair. It's actually longer than that here. And this one, right? So if you bounce to here, then you read out 300 base pair here. Okay, so let's assume that you read out from here to here. Okay, so what is the first position? Here? No, this is the first position. Okay, and second position, third position, 300 base pair position, right? So this is how the relationship between the library, fragment, and adapters and reads and each cycle position. You have to remember these things, otherwise you cannot understand entirely afterwards, right? So this is the key things in this lecture. Okay, so let's move on to Illumina sequencing summary. Illumina sequencing technology is the super term. Within that, Illumina has different machines. So it has the HiSig 10, NextSig. It's like a iPhone, iPhone Pro or iPhone SE, something like that. And they have the different, throughput, different price, and different, advantages, right? So these things you can read in their commercial websites. And Illumina sequencing and next-gen sequencing, they have specific format, which is the single-end paired-end sequencing. So as I mentioned previous slides, once you have the library, like in here, you have an adapters on here, right? Adapter here, and this is the DNA fragment. Then you can read this position, you can read this position. If you read just one time, we call this is the single end read, right? One end and just one thing. So it's a single and end. And read is this one, okay? But if you read both reads, so if you read one and you flip out that, then if you read the second thing, then you call reads two. And those kind of sequencing we call paired end read, right? So you have the information in here, you have the information in here. So you can basically have two different types. One is a single end and the other one is a paired end sequencing. And paired end sequencing is generally much better than the single end reads. So these days, people not really using single end reads, but some technology still use it. But paired end sequencing is more universal. You can use in your data set. Okay, so this is the scheme of that and that information too. Okay, so as I mentioned that the NGS is basically the short read sequencing. So the newer things, people advanced in this field, they introduced the long read sequencing. One of the two technology we can think about the long read sequencing is the PacBio. Pacific Bioscience, and they use the smart bell template, which is the two hairpin adapters at the end of that. Then you use the double strand like in here. Then you put this information into the wall and you do the fluorescent sequencing. So it's a little bit different chemistry than Illumina, but it will give you much longer reads than Illumina one. Illumina one is around like a 300 base pair to 200 base pair, but PacBio sequencing, they sequence up to 70,000 base pairs, which is super longer than Illumina one, right? So if you read out this information, then you can think that why we not use just the PacBio sequencing, not the Illumina sequencing, right? This is much longer and looks like better than that, and coverage is good. But why? Because it's much expensive than Illumina. Still today, right? But maybe later, year later, or 2 year later, or 3 year later, PacBio sequencing is much cheaper than that, then everyone can use it. So we don't know that, right? Illumina sequencing, if you sequence one person, just yourself, it's around, I can say $700 USD. PacBio sequencing is like a 5,000 USD. So much, expensive than that. So people always try to think about like a trade-off between the data quality, data length, and price, right? Okay, just for your information. Another technology is Oxford Nanopore. It also used the hairpin templates and it used the motor protein to read that information. But different thing is they not measuring fluorescent, they're measuring the current shift, right? So they have unique current to ATGC and they get that information, then they decipher current to transfer into the sequence information. And nanopore sequencing also can give you longer reads than Illumina, and also is quite similar to PacBio. I mean, the example is not that correct. It can up to like I heard that it's around like a million base pair, I guess. It's quite useful. But most useful things about the nanopore sequencing is this one. It's portable. Okay. So fluorescent, as I mentioned in Illumina, is like a 2-finger size, right? And this 2-finger size fluorescent, they get it into the Illumina machine. And Illumina machine is like a fridge size. It's super big, right? But nanopore, fluorescent, and sequencing machine, the same. So this is also like a 2-finger size. And this is the thing you can do the flow cell preparation sequencing together, right? So this is like a USB size, right? So that's why this person brought nanopore into the space, right? ISS, right? And they measure different biological materials throughout this nanopore sequencing. So they measure the Drosophila in a astro situation or plant, I guess. I don't think monkey, those poor thing. But anyway, those animals and plants, they measure it through here. Or nanopore sequencer is quite portable. So they brought to the Amazon or, quite, distant locations in Brazil when the Zika virus outbreak, and they sequenced there directly, and they opened that information to the scientific field, and they got to know about how Zika virus is happening, right? So this is also advanced we can make by nanopore sequencing. And also nanopore sequencing gives you much better finding in terms of the deletion, right? So you can see the deletion sharply, you can see on here. Illumina sequencing is a little bit dirty to see these regions. So this is also advantage of the longer reads. So that's why we can see much higher resolution in terms of that information. Okay, so on the next steps, we have to think about the base calling process, right? So how we can find the sequence from the sequence data. As I mentioned, that Illumina sequencing gives you fluorescent information, then that information has to convert into the text information, into ATGC, and nanopore is the current information that has to change into the text information too. So those information, it's done by base calling algorithm implemented in the each machine. So you don't have to think about that. But you have to know the raw signals made by machine is either fluorescent image or current, right? So how these things is related to your information once you get the DNA extracted from someone, that someone has the age sequence from mother and father, like in here. Then if you put it into sequencing machine, that information changed to the text information like in here. This is like a crazy character, but this is the thing you're going to use in the genomic analysis. And that information is not just a few lines, it's going to be like a 100 million lines in your data, right? And this is just for one person, right? This is the crazy thing. So if you look like 1,000 people, if you look like 100,000 people, if you look at 1,000,000 people, the amount of data set is immense, right? So, I mean, I just saying, so, get to feel about the sheer amount of data set, right? So if you look at here, so a dad and mom give you reference allele, then that information that will convert into like this base calling data. And if you look at the position, you have the many reads covered by either your mom or father, right? Mom or dad, right? And that information like in here. If you have the homozygous genotype, then CC, that information also mapping like in here. And if you have the CT heterozygous information, that will be like in here, right? Okay, and this is basically how the base calling is working. And the next one, the final thing is what is the types of different genomic sequencing, right? So we can have the 3 different genomic sequencing, looking at the DNA sequence. I'm not talking about the RNA sequencing or epigenome. We might cover in the later section, not on this time. If you look at the genome sequencing, the DNA sequencing, then you can have whole genome sequencing. Literally, you look at the whole genome, right? You're looking at all the nucleotide in the genome. Or you can looking at the exon region, which is the exome is the region which contain the coding sequence that can produce the protein coding gene. That is the 1% of the genome. So this is much tinier information, and you can directly find the mutation affecting on the protein production, right? Or you can do target sequencing. You can only read out certain area of your interest. So if you want to looking at the cancer region, then you're looking at the target sequencing. And after that, you can compare between the NGS versus microarray. Microarray, as I mentioned last time, you can only looking at the SNPs we know previously, but NGS is you don't need that. You can just directly read out all the nucleotides. That's the good thing. And this is bit schematic things between the exome sequencing and target sequencing. So if you use the exome sequencing, you can basically use the biotinib target probe. Then you pull out the regions of your interest, either it's a coding region or cancer genes, whatever. Then you can pull out that information, then you put it into the library. So that's how exome or target sequencing is working. Okay, so this is the thing we covered the basic information about next generation sequencing, and next time we move on to which data format we can think about, we can generate from the NGS and how we can find the genetic variants, alleles out of these NGS datasets. So see you next time.